Hi, my name is Gemma Gordon. I'm a PhD student at the University of Oxford, and today I'll be talking about a recent project which was a comparison of the binding sites of antibodies and single-domain antibodies. Firstly, to provide some context, we know that antibodies are currently the largest class of therapeutic, however there's increasing interest in single-domain antibodies. These two antibody formats differ in the structures of their binding sites. Most notably, single-domain antibodies have half the number of CDR loops that conventional antibodies do. So here we ask whether antibodies and single-domain antibodies differ in how they interact with the antigens that they bind to, and whether, given their structural differences, they're suited to different types of epitopes. To compare our two antibody formats, we use publicly available sequences and structural data, and we define the paratopes and epitopes of our structures using the interactions occurring between residues, using the software Arpeggio for this annotation. When we compare firstly the paratopes of antibodies and single-domain antibodies, we find that, as we might expect, overall they're quite different. For example, in agreement with previous work, the CDRH3 is longer in single-domain antibodies. It's been hypothesized that this allows single-domain antibodies to target more cryptic epitopes that are less accessible to conventional antibodies. We also find that the paratopes of single-domain antibodies have fewer residues on average than those of conventional antibodies. Next, when we compared the epitopes, we found that antibodies and single-domain antibodies are able to target epitopes of similar size, despite the fact that we found paratopes of single-domain antibodies to be smaller. I'll come back to this result in a couple of slides when we compare the interactions occurring in these complexes. We also observed for the paratopes that single-domain antibodies have longer CDRH3 loops, and we find that single-domain antibodies have significantly less accessible epitopes than conventional antibodies. However, this difference is only marginal for our datasets. Further to this, we find no correlation between the length of the CDRH3 loop and epitope accessibility. We propose that the ability of antibodies and single-domain antibodies to target epitopes of similar accessibility is due to differences in the orientation and compaction of their CDRH3 loops. So we find that although the CDRH3 in single-domain antibodies tends to be longer, this may not translate to greater reach in all cases due to compaction of the loop against the rest of the VH domain rather than the loop extending out into space. So now, moving on to comparing the interactions of these two antibody modalities, we saw earlier that single-domain antibodies have smaller paratopes, but are able to target epitopes of similar size to conventional antibodies. On comparison of the number of interactions, we see that single-domain antibodies may be able to achieve this due to their paratopes initiating more interactions per residue than conventional antibodies. Lastly, we note that framework residues are of increased importance in the paratopes of single-domain antibodies, but these are often conserved, meaning that single-domain antibodies may incorporate less specific interactions into their binding sites. So, in conclusion, we find that single-domain antibodies have smaller paratopes than conventional antibodies, but they're able to target epitopes of a similar size. And to achieve this, the paratopes of single-domain antibodies are contributing more interactions per residue than those of antibodies. Secondly, we find that conserved framework residues are of increased importance in single-domain antibody paratopes, and this suggests that they're using more non-specific interactions to achieve comparable binding affinity. Thirdly, for our datasets, the epitopes targeted by single-domain antibodies are only marginally less accessible than those of conventional antibodies, and we think this can be explained by differences in the orientation and compaction of the CDRH3 loops. Overall, our results have important implications for the engineering and humanization of single-domain antibodies for therapeutic use, as well as choosing which modality would be best for a particular epitope. So overall, despite there being differences in the paratopes of antibodies and single-domain antibodies, the epitopes that they target are not as easily distinguishable, and we suggest that this is due to differences in how these two modalities interact with their targets. So finally, I'd just like to thank my co-authors from the Oxford Protein Informatics group and from Twist Bioscience, everyone else who contributed to the project, and thank you for listening.